Good morning, everyone, and I use that term loosely because, as you know, classes have been canceled, and this video will be uploaded to the class website. It's the only way, I guess, we have of interacting today in light of the coronavirus. But I don't like lecturing to an empty classroom, so I've decided to give this lecture today to this cheese Danish. So today we're going to talk about the CRISPR decision, and I don't know about the cheese Danish, but I think that everyone here is going to find that the CRISPR decision, which involves the patenting of a gene editing technique that I think everybody's heard about, especially everybody at MIT has heard about. I think this CRISPR decision in many ways wraps up the discussions that we've been having because it really does illustrate almost all of the concepts that we've been talking about, from what is patentable to the importance of claims and interference, and how interference cases are won and lost. And so with that, let me sort of jump into CRISPR with a little history of this gene editing technique. I think everybody's heard about it. Of course, it's famous for the lawsuit that existed between the Broad Institute here at MIT and Stanford in California. And CRISPR, of course, has been known or has been called the greatest biotech innovation of all time. I may not go that far because of the things that we've talked about, even when it comes to this type of gene editing technique. Science marches on, innovation occurs, and there are workarounds even for CRISPR, and even though this technology is relatively new. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. CRISPR are basically segments of prokaryotic DNA containing short repetitive base sequences. In a palindromic repeat, the sequence of nucleotides is the same in both directions. Each repetition is followed by short segments of spacer DNA from previous exposures to foreign DNA, like a virus attacking a bacterium. And that's really what the scientists that were responsible for this breakthrough, that's what they did. They studied the way bacterium react to various viruses. And they adopted this gene editing technique from what bacterium have been doing in response to viruses. This is how bacteria protect themselves from viruses and how they've been doing it for millions of years. Finally, some scientists came along who've decided that this gene editing technique that bacteria have developed a natural ability to perform has tremendous potential when it comes to the curing of diseases or even the creation of super agricultural crops. The use of CRISPR Cas9 RNA complex for gene editing was the choice of the American Academy of Sciences Invention of the Year in 2015. In any event, CRISPR is a form of acquired immunity which was adapted from a naturally occurring genome editing system in common bacteria. The bacteria capture snippets of DNA from invading viruses and use them to create DNA segments known as CRISPR arrays. The CRISPR arrays allow the bacteria to remember the virus that it had attacked it before, and when it acquires the virus or is attacked again by the virus, the bacteria remember that the DNA of that virus, and they use that array to attack the virus and thereby protect themselves. They use the enzyme, the Cas9 enzyme, to cut the DNA apart and rendering the virus not harmful to the bacterium. Obviously, if you can apply that sort of gene editing to human beings, it has enormous potential when it comes to the cure of diseases like cancer. And of course, this is what has been seized on by scientists from coast to coast. So the question for us becomes, can you patent a naturally occurring substance or not? Because really, when it comes down to it, bacterium have been doing this for millions of years. Is that the kind of thing that can be patented? Remember, we talked about what's patentable. This comes from the statute. Whoever invents or discovers any new and useful process, machine, manufacture, or composition of matter, or any new and useful improvement thereof, may obtain a patent, therefore, subject to the conditions and requirements of this title. So the question is, does a gene editing technique such as CRISPR qualify under this definition of what is patentable? The term invents or discovers requires that the patentee be the actual creator and not just the first person to come upon something that already exists. And the use of the word discovers is actually, I think, unfortunate. But it makes it seem as though a person can get a patent on something that's naturally occurring, when we have already discussed, of course, that one cannot, even if you're the first person to find it. But the courts have consistently held that while one cannot get a patent on laws of nature or natural phenomenon, even if no one has discovered it before, that's why you can't patent the speed of light or gravity, but one can get a patent on a new use for a law of nature or natural phenomenon, since any new invention has to follow the laws of nature. So just because it follows the laws of nature doesn't mean that it's not patentable. And since bacterium have not been curing cancer in human beings, this gene editing technique, when applied to creatures, multicell creatures like human beings, is of course the application of a newly discovered natural phenomenon in a new method, in a new application. So it may involve a natural substance which has never been seen or isolated before, and in this case is patentable because the application is novel. We talked about the requirements of novelty and usefulness. Well, this is a natural phenomenon, but it's being applied in a unique or novel way in order to produce a result or to do something, and therefore it satisfies the requirements of what is patentable under Title 35. So I want to talk a second about how actually CRISPR works. The system works similarly in a lab as it does in bacterium. Researchers create a small piece of RNA with a short guide sequence that attaches to a specific target sequence of DNA in a genome. And then the RNA also binds with the CAS enzyme. Just like with the bacterium, in the laboratory, what scientists have done is use the CAS9 enzyme to literally cut the sequence of DNA apart. And so although the enzyme is used most often, the CAS9 enzyme is used most often, other examples of enzymes like CPF1 have been used. And as I said, since this technology was introduced, there have been all kinds of workarounds, different enzymes used. Everyone is basically trying to mimic what bacterium have been doing for millions of years, using various enzymes to first to identify and remember the target virus, and using this enzyme essentially to cut the virus up, the DNA sequence and the virus apart. And once the DNA is cut, researchers use the cell's own DNA repair machinery to add or delete pieces of genetic material or to make changes to the DNA by replacing an existing segment with something customized. And this is where the therapeutic purpose behind the CRISPR gene editing technology is involved. So, and all of this began back in 2012. Well, actually, it began before that, but in 2012, the University of California, in fact, Jennifer Doudna and others published a scientific paper on CRISPR called a programmable dual RNA-guided DNA endonuclease in adaptive bacterium immunity. This was really the beginning of the whole CRISPR story. This paper was such a breakthrough that everybody almost immediately seized upon the potential of it because they could see the therapeutic applications of this. And in this paper, Professor Dudna showed that the gene editing technology can be used to cut DNA in a test tube of targeted sites. And these advances fueled efforts to edit genomes with the modified CRISPR enzyme. Later, what Professor Dudna did is filed a patent application. But key to this is what she was able to demonstrate. And that is that the technology can be used to cut DNA in a test tube at targeted sites. That actually will be the most important part of what eventually turned into a very big and expensive piece of litigation. So what they did is they create a small piece of RNA with a short guide that attaches to a specific target sequence. The RNA also binds to the enzyme. And as in bacteria, the modified RNA is used to recognize the DNA sequence. And the enzyme cuts the DNA at the targeted location. Actually, it's a pretty simple natural process. And as I said, bacteria have been using it for years to protect themselves against various viruses. So a CRISPR spacer sequences are transcribed into short RNA sequences capable of guiding the system to matching sequences of DNA. And when the target DNA is found, the enzyme, one of the enzymes produced by the CRISPR system, binds to the DNA, cuts it, shutting off the targeted gene, or if you will, turning off the cancer cell so that it can't replicate, it can't invade the host. Now, this is all research that was actually based upon something that was discovered at Osaka University back in 1987, but they didn't understand what they were noticing. The organization of these repeats was unusual. It was first noticed by Professor Yoshizumi, but they really didn't understand the import of what they were observing. Again, this is not something that what they were noticing here was the natural phenomenon. And as we've discussed, natural phenomenon are not patentable. So the natural phenomenon was first noticed back in 1987, but what to do with it and how to apply it, that took Professor Dr. Dudna in 2013. So where did the dispute begin? In 2013, in another scientific journal, an MIT bioengineer by the name of Fang Zhang and his team over at the Broad Institute developed a CRISPR system that edited genes in living cells of mice and human beings. And this was a tremendous advance. Up until then, the technique had only been demonstrated with single-cell creatures in a test tube. And this advance by the Broad Institute, demonstrating the technology on living cells of higher animals, mice in this case, and human beings, represented a tremendous advance in the technology. Not in the phenomenon. The phenomenon had been something people were aware of since 1987. But the application of the phenomenon, especially in higher animals, was something entirely new. And so when the Broad filed its patent application, the Broad asked that it be fast-tracked. And we've talked about that. It costs a little bit more, but you can move things more quickly in the USPTO if you fast-track. So if you picture the point in time, in 2012, you have the University of California filing an application with the USPTO for their CRISPR gene editing technology. And then 1 year later, you have the Broad Institute simultaneously filing a patent application with the USPTO for their gene editing technology. And they're both going on parallel tracks at the USPTO. And as we know, sometimes this takes 2 and 3 years. It was a good thing. It was insightful for the Broad to fast track their application. And one of the reasons they did that is because they had confidence in being able to demonstrate the efficacy of the technology for this particular application. They knew, of course, that other universities, research universities, were working on this technology, and it was a race for something potentially incredibly lucrative. So the University of California, which was waiting for its decision from the USPTO on its own patent application, filed an interference proceeding against the Broad and tried to essentially to get their patent application canceled by the USPTO. And the reason is the University of California said, look, we already have discovered this technology. We've already filed a patent for this technology. The Broad's application for a patent interferes with our application, and the USPTO should dismiss it. In February 2017, so we're actually moving 4 years later. It took 4 years. The USPTO ruled that the Broad patents, with claims covering the application of the technology for human beings, was distinct from the invention claimed by the University of California. The research done by the University of California had only demonstrated the technology in prokaryotic organisms, single-cell bacterium and the like. Prokaryotic organisms are microscopic single-celled organisms that has neither a distinct nucleus with a membrane nor other specialized organelles. Prokaryotics include the bacteria and cyanobacteria. So that's a long way from mice and human beings. In fact, it's probably a long way from this cheese Danish. It's the simplest form of life on planet Earth. And the only thing that the University of California could do is that they could show that their gene editing technology worked with these types of organisms. Eukaryotic organisms, by contrast, are composed of multicells containing a visibly evident nuclei and include complex organisms such as humans and plants. So you and I are eukaryotic organisms. And the University of California was not able to demonstrate this technology had any efficacy in eukaryotic organisms, just prokaryotic organisms. So editing of eukaryotic cells is what scientists led by the Broad Institute did. University of California researchers never demonstrated this technology with eukaryotic organisms. And so the USPTO ruled it is undisputed that the University of California does not report the results of experiments using this system in eukaryotic cell. And that was the truth, because when they filed their claims, and I'll get into it, the scientists, including Dr. Dudna, gave a number of high-profile interviews to the New York Times and scientific journals, where they talked about their technology and what it could and could not do in great detail. And that later on came back to haunt them. So when it filed for its patents in 2012, the University of California and the co-authors of the paper asked for patents covering the CRISPR genome editing in any setting, including eukaryotic cells and other cell types. Now remember, we talked about how claims are everything when it comes to patent applications, because a claim is like the fence, the boundary fence that defines the intellectual property that you own. And as with most people that file patent applications, I imagine they sat in a lawyer's office someplace in California and came up with this phrase to include in the claims section of their application. But this was a stretch because they had not actually demonstrated the technology in eukaryotic organisms. But they said in their application that that their claims should include that, even though they had not demonstrated the efficacy in those organisms. The patent application, remember this being a new form of gene editing technology, the Patent and Trademark Office was just coming up to speed with this technology. I'm sure if the Broad had not come along, that broadly written claim would have slipped by, and the University of California would have had a monopoly on this technology worldwide. But that's not the way it worked out for them. In its interference claim, the University of California argued that their patents and the one applied for by the Broad overlapped, because without UC Berkeley's pioneering work on CRISPR, the Broad's work could never have taken place. The Broad argued that its patents on the use of this CRISPR editing technology when it came to animal and human cells didn't overlap UC Berkeley's application. So what you have here is the UC Berkeley saying, look, you know, what the Broad has done is really not very innovative. It's not novel. It doesn't satisfy the requirement of novelty. It's an obvious advance or an obvious development of our technology, and it should not be separately recognized. The Broad, on the other hand, saying, wait a minute, hold on. Our advance is very significant. It is novel. It is revolutionary. It is new. And there is sufficient daylight between what the University of California has done and what we have done to warrant a separate patent. And how do they know this? You know, how does the Broad and the University of California have this fight in front of the USPTO? What do they provide the USPTO? Well, in the trial that takes place before the USPTO, the interference trial in this case, the Broad brought in scientists to describe to the administrative judges that make these decisions the nature of the technology that they had developed and its efficacy in animals and humans. And the University of California brought in their scientists armed with their research papers saying that their technology was unique and that the Broad's was really just a natural or sort of common sense application of what they had already discovered, and that there was really no daylight between the two. So the question is whether or not it was an obvious sort of development of something that had already been patented, or was this truly innovation? And what they argued about is not just who invented CRISPR first, because, by the way, when this case took place, it was back in 2012 and 2013, which was before the America Invents Act was adopted, and we became a first-to-file country. They were dealing with the old laws, the old rules, which also said, you know, we needed to find out who was first to invent. So they weren't just talking about who was first to invent, but also who first unveiled key aspects of the technology. University of California claimed that after the publication of her paper, anyone could have applied the technique to edit eukaryotic cells, the cells of animals and people. They argued that this was an obvious development. It wasn't real innovation. And the Broad argued that it was a huge inventive leap to actually apply this technology to complex organisms. California argued that 6 other research groups were working on using the CRISPR technology in eukaryotic cells. 6 months after they had published their paper in 2012, they were using the same system that they had used, and its attorneys argued there's no special sauce here. That's actually a quote from the brief. what they're basically saying is once we broke the ground on this technology, then there were research groups working all throughout the country standing on our shoulders trying to apply this to eukaryotic cells, and that there was no special innovation or insight to the Broad and what these other places were doing, because they were saying everybody was working on this. And actually that was true. Everybody was working on using this gene editing technology in eukaryotic cells. The Broad just happened to be the first to apply for a patent, and they were smart enough to fast track it. So they really got their foot in the door. It shows how sometimes, you know, the winner is the one who wins the race to the USPTO. The Broad argued that that his team had a reasonable expectation of success, and they used conventional methods, meaning that the Broad didn't do anything particularly inventive to successfully use the CRISPR technology. And these are, by the way, these are terms of art in the law. A reasonable expectation of success is a term that the Supreme Court uses to determine whether or not this is an obvious advance or whether or not this is an innovation. If when you patent a technology, there is a reasonable expectation of success that the technology can be applied in other areas which haven't necessarily been demonstrated, then it's probably an obvious advance and not an innovation. So the question is, hope of success or reasonable expectation of success? The Broad argued that even though other groups were trying to use the CRISPR system in eukaryotic cells, no one before them was able to make the system work. And so there was no hope of success. But so while there was hope of success, there's a big difference between that and reasonable expectation. So what the Broad said is, yeah, everybody's working on this. We weren't the first to think it up, but we were the first to demonstrate its efficacy. So while there may have been hope with these various groups, whether there's 6 or 600 groups working on it, we were the ones that were able to do it. And there's a big difference between hope and reasonable expectation of success. So determining who owns that intellectual property basically means determining who's going to be rich, the Broad or University of California. This gene editing technology has potential applications in everything from medicine to agriculture. A patent holder, of course, will impose hefty licensing fees, both when it comes to research universities and institutions that are developing therapeutic methods. And can you imagine what Novartis or Pfizer or any of the companies over here in East Cambridge would pay for exclusive rights to this gene editing technology that has the potential to cure cancer or even develop super crops. So the company that owns this technology, the company that owns the patent on this, is going to make an awful lot of money. So what happens now? Who wins and who loses? Actually, this case actually has come to a conclusion I can tell you about. What happened in this case is the Broad received a patent for its CRISPR gene editing technology in eukaryotic cells. The patents that came from the University of California, the ones that they filed in 2012, but which were held up by the interference proceeding, were granted to the University of California, but on a limited scale. And it was only for prokaryotic organisms, not eukaryotic organisms. The TTAB decision, and TTAB stands for Trial and Appeal Board. That's the arm of the USPTO that tries cases administratively. When you bring an interference action, you can either bring an interference action at the USPTO, because they have judges and administrative personnel for dealing with these disputes, or you can walk over to the United States District Court and file a private or civil lawsuit there. If you file a civil lawsuit in the federal courts, the proceedings before the USPTO are stayed. since it's considered an inferior jurisdiction to the United States District Court, just like the United States District Court is considered an inferior jurisdiction to the United States Appeals Court, which is considered an inferior jurisdiction to the United States Supreme Court. So they had a choice. They could bring they could bring their interference action at the USPTO before the Trademark or the Trial and Appeal Board, or they could have brought it in federal court. But because the expertise was available at the USPTO, I think the strategic decision was to go with the decision, or go with the filing of the interference case with the USPTO, and that's literally what they did. The TTAB decision offered, though, a little olive branch to the University of California, stating that an earlier disclosure of a genus does not necessarily prevent patenting a species member of the genus, suggesting that a larger category of something can be patented separately from a subset of it. What does that mean? Well, what it means in this particular case is that if the University of California had been able to demonstrate the efficacy of this technology in eukaryotic cells, they probably would have gotten the patent. They would have been able to, the USPTO would have approved their very broad claim. But in this case, because they weren't able to demonstrate it, they lost. I like to think of it as green tennis balls. Any company that wants to use CRISPR to develop human therapies may need not only to license the Broad's patents for eukaryotic cells, but also those from the University of California. The University of California patent would be on any use. The Broad would be only for eukaryotics. So if somebody wanted to do this technology or do this research in eukaryotics, they would potentially be required to license the technology from both. And that's where the green tennis ball comes in. In patent law, it's possible to get a patent on all tennis balls, and for someone else to get a patent on green tennis balls. The technology owned by the University of California is all tennis balls. The technology owned by the Broad Institute for Eukaryotic Cells is for green tennis ball. All tennis balls are all cells. Green tennis balls are eukaryotic cells. So you can get a broad patent on a technology, but not own every application of that technology. And that's exactly what happened here. In the judge's decision that there was no interference, in fact, meant that CRISPR patents issued to the Broad covered different inventions than those in the patents for the University of California applied for. And crucially, the decision was that it was not obvious to go from the work that the University of California did to what the people at the Broad Institute did. By the way, a lesson to learn from that case is the old saying, loose lips sink ships. The reason the University of California lost their case is because the inventors of the technology gave very, very detailed statements to reporters, which led the U.S. Patent and Trademark Office to conclude that there was no overlap. The USPTO even cited an interview that the inventor gave to a scientific journal where she stated, "We weren't sure if CRISPR-Cas9 would work in eukaryotics, plant and animal cells." She said it, and the USPTO hung her out to dry on the statements that she had made to the press. And that's why loose lips sometimes sink ships. I think that the scientists were so excited about what they had done that they went out there and really were acting like scientists, trying to not only trumpet their discovery, but also share the results of their research. But by being so forthcoming, the researchers at the University of California ended up sinking their own ship. Based mostly on her candid comments, the USPTO concluded that inventors themselves were uncertain about making CRISPR work in human cells. And I actually saw the cross-examination of the University of California's experts in this case. And so, University of California brought in their expert witnesses, and their expert witnesses said that, hey, this is an obvious advancement. This is not, what the Broad has done is really not that innovative. And then on cross-examination, they were confronted with the statements of the researchers from the University of California, and they had a very hard time reconciling their statements with the statements made by the actual inventors. Those quotes by the inventors were basically the end of the University of California's case. So the question then becomes, can scientists afford to be honest? Based mostly upon the inventors' candid comments in the press, the USPTO concluded that they weren't entitled to a patent on the eukaryotic application of this technology. And that really can send sort of a mixed message to scientists. Do we need to coach scientists to not be honest about their technology? And I think the answer to that is, for the most part, yes. If in particular in this case, if you're unsure about whether or not you can demonstrate the efficacy of your technology, and you've made a claim that you can, well, don't go out and tell the New York Times that you can't, because ultimately you may be sitting on the witness stand, as they did, as their experts did, and be confronted by these statements that you've made, which are direct opposite to the position you're trying to maintain in the litigation. I think the legal fees in that case topped $50 million ultimately. That's how much money they were willing to wager on legal fees in order to wrestle this technology away from one another. And the funny thing is, what they really should have done is sat down, especially for $25 or $50 million, whatever actually the number was, and work out a solution, an amicable solution to this. If the University of California and the Broad Institute had wanted to, I think that they could have both come up with a way that they could have shared the technology, joined together and licensed it to other parties. But of course, while they fought, what happened? Remember what happened when it came to graphene. Those the scientists that won the Nobel Prize, and they were able to isolate a one-atom thick matrix of graphene using Scotch tape, and that was the end of the world as far as everyone is concerned. They had patented the technology. The development of graphene has ceased, and as we all know, that just didn't happen. What happened in this case, is the same thing that happened with graphene, and that is that other organizations all across the country developed workarounds to this using different enzymes. So all they did is they went back to the bacteria that the University of California had studied, and they said, well, how else does the bacteria do this? And they found that other enzymes besides the CAS9 enzyme they were able to produce the same results in eukaryotic cells, cell organisms. And so rather than paying the Broad and the University of California, science inexorably marched on, and innovation took over, and they came up with workarounds to the technology. So now if you want to, if you're a big pharmaceutical company here in East Cambridge and you want to use this gene editing technology on a therapeutic method that you have in mind, you might license your technology from University of California and the Broad, or you may either develop your own or license it from someone else using a different enzyme or different method. The bottom line is that science and innovation always trumps. They'll always find, at least in my experience, a workaround to these things. And if you have $50 million to spend, probably you'd be better off spending it on anything other than lawyers, because while you fight it out, somebody is sitting in a classroom here at MIT or someplace else trying to figure out a way around the technology that you're fighting over, which is a lesson I hope that the Broad and the University of California have taken to heart. They really should have settled rather than fight this thing out. The postscript is that in September of 2018, the U.S. Court of Appeals awarded the pivotal intellectual property to the Broad Institute and Harvard and Cambridge, upholding the decision by the USPTO. So ultimately, when the University of California didn't like the decision by the USPTO, they did what they have a right to do, which is appeal to the First Circuit Court of Appeals, which is the court that has been created strictly for intellectual property, and they didn't do any better there. So the Broad Institute owns the technology for the CAS9 enzyme gene editing technology for eukaryotic cells. If you want to work on other cells, I guess you'll have to license from the University of California, or come up with your own innovation, which many have. So that's it. One last postscript. I wanted to let you know that our hypothetical student from the famous university in Cambridge, who invented a hydrogen generating machine, has prevailed, and the patents have been issued, and the way has been cleared for another millionaire inventor here at MIT. But the good news is that hypothetical case study that I talked to you about on the beginning of school 6 weeks ago has worked out in favor of the students, which is proof that sometimes the good guys win, or cheese Danish, depending upon the circumstances. Thanks for watching the lecture. I hope this has been helpful to you. I have, by the way, course evaluations that will be available through Professor Eager's office, or we'll put them up online. I would value any insight that you have, whether it has to do with my choice of bow ties or the lectures. But any input that you can give would be much appreciated, especially by students who will be coming next year. I want to make this course better for everybody, and I can use your insights, whatever they may be. So thanks for watching, and I'll let you know if you have any questions or any concerns. You contact me. There we go.